righty. Okay, welcome everybody. Today we're going to continue our discussion of viruses and prions. Okay, and specifically, today we're going to talk about replication. Okay, here is the overview of what we're going to do today. I'm going to go over the learning objectives with you. I'm going to open it up to questions. Then we'll talk about bacterial phage, replication. Then we'll go into animal virus replication, and then we'll talk about what to do for next time. Okay, so this chapter helps us move toward meeting the following objectives: being able to define the key role of evolution as it applies to microbiology, identifying microbial structures and connecting the structures to their functions, identifying key agents and processes of microbial information transfer, identifying pivotal components of microbial systems important to human health, and being able to analyze and describe the impact of microorganisms. All right, any questions? Okay, if you think of any, let me know. I am happy to stop and answer them. Okay. All right. The first topic that we're going to cover is bacterial phage replication. So let's go to the whiteboard. All right, first off, bacterial phages are bacterial viruses. And the reason why they're not called viruses is because the folks working on bacterial viruses were working separately from the folks working on animal viruses. And so they ended up naming them different things. Okay. So bacterial phages, okay, are bacterial viruses. And they have been exceptionally useful in our learning about viruses and our study of viruses, because bacteria are very easy to keep in the lab. Okay? So we're going to talk about how these viruses replicate, okay, or make more of themselves. Okay. So there are 5 steps in bacterial phage, or we often abbreviate it as just phage replication. Okay. Remember, viruses are not alive. They don't have some of the necessary structures to have them be included in with cells as living organisms. Okay. And so they're not alive, so they can't reproduce, but they can make more of themselves, like little robots. So there are 5 steps, and we're going to go through all of them. The first one is attachment. The next one is penetration. Okay. And bacterial phages, we have another step that's combined with this. When we get to animal viruses, we will see that these are separate steps. Uncoating. Okay. Then we have synthesis. Then assembly. And then release. Okay, so we're going to go through all 5 of these steps. Okay. I'm going to clear this and let's get a standard bacterial cell up here. Okay. So this is the cell membrane. And then since we usually study bacterial phages and E. coli, I'm going to make a gram-negative cell. So this is the thin peptidoglycan cell wall. And then on the outside, we have an outer membrane. Okay. And then on the inside, we have a circular bacterial chromosome. Okay. And it is DNA. Okay. Also inside of the cell, we have ribosomes. Okay. Got a bunch of ribosomes that in the normal process of things, okay, the DNA chromosome is transcribed, okay, and those mRNAs go to the ribosomes, and we make bacterial proteins that proceed to make other stuff, okay? All right, so in comes our bacterial phage, okay? And I'm going to draw one that, I'll be honest, is simply entertaining for me to draw. Okay, and it is a DNA virus. Okay, and we have the capsid. Remember, if we don't have some sort of genome and a capsid, okay, and spikes, although in this, instead of calling them spikes, we call them tail fibers, or to me, they look like plates. Okay. So here is my phage. So the first thing it has to do is attach. In other words, it has to grab a hold of the host cell. Okay. So it just happens to bump into the host cell. And because the spikes or the tail fibers recognize proteins on the surface of the cell, they're able to grab a hold. Okay. So attachment is grabbing a hold. Okay. Okay, now the next thing that needs to happen is this DNA has to get inside of the cell. It can't do anything out here. Okay, this is just a way of protecting the genome between cells. The real goal is to get in. Okay, so we call that penetration. Okay, so that's getting inside of the cell. Okay. And then uncoding is getting the DNA or the RNA outside of the capsid. Okay, now this happens in one step. Okay. In bacterial phages, penetration and uncoding happens in one step. Okay. What happens is this sheath, okay, gets pulled down, and there's these almost like blades, okay, that punch through the cell wall, okay, and the phage genome is injected into the host cell, okay. So the DNA was able to get outside of the capsid and into the cell in one step, okay. All right. So now that the DNA is inside of the cell, it convinces the cell, okay, that this DNA needs to get transcribed, okay, before the bacterial chromosome. Okay. Now, the phage that I'm using it as an example, okay, does this two ways. Okay. One, there are signals, promoters on this DNA that are more attractive, okay, to RNA polymerase than on the bacterial chromosome. Okay? And the other thing that it does is it releases an enzyme that chops up the bacterial chromosome. Okay. So basically it destroys the DNA of the cell. Okay? Also, in the process of getting in, it has released another enzyme that is proceeding to punch holes, okay, through the cell membrane. Okay? So once penetration and uncoating happens, there's only a certain amount of time that the phage has to make more of itself before the cell dies. Okay? So it's got holes that are being punched in its cell wall, and it no longer has a chromosome, so it can't make any more of its proteins. So it's on its way out. The cell is going to die. Okay. So the next thing that the virus has to do is make parts. Okay. So we call this synthesis. Okay. Okay, so remember, these are kind of like little robots. They're not animals. Okay, they are not alive. They're not bacteria. Okay. It's kind of like they've got to make their parts and they get snapped together like Lego pieces, just like a machine. Okay. So the first thing that needs to happen is we get some transcription going on. Okay, so we make some mRNA that get clamped onto by the ribosomes, okay, and the ribosomes proceed to make viral proteins, okay. So we start making capsid parts, okay, we'll start making some cell fibers, okay, and we also need to make more of the genome, okay, because when we snap all these together, there better be viral genome in there, okay, so we need to make more of the viral DNA. Okay. That's kind of a scary double helix. Let's make one that looks a little more like DNA. Okay. Now, just for the sake of space, we're going to say we're going to make 3 of these guys. Okay. That's not very many. Usually, with bacterial phages, they make between 200 to 2,000. Okay. During the time before they've killed the bacterium and the bacterium pox. Okay? So once we get all of our parts synthesized, okay, we've got the parts of the capsid, we've got the sheath, okay, so we've got the parts. Now we need to snap them together. Okay, so that is putting it together. And because they're not alive, there are no baby viruses, there are no baby phages. Okay, they snap together like machines. Okay, so I'm going to put the capsids together with their version of a spike. And let's put another one right here. In fact, you can buy parts of phage, okay, get them through the mail, put them all in one container, shake them up, and they will self-assemble. Okay, so they self-assemble within the bacterium, okay, and we put DNA in, okay. And then remember that this enzyme, which by the way is a lysozyme, I mention it because you don't need to know it on the test that covers this chapter, but we will be talking about it later, so I want you to have heard the name. Okay. Oh, let's see, there's no S in it. It's just lysozyme. Okay. So has been punching holes in the cell membrane. and eventually the cell pops, okay? And it releases the virus particles out into the environment. So I'm going to get rid of my cell wall there, okay? And my phages are released, okay? And they go off looking for another bacterium, another host, okay? And we start the process all over again. Okay, questions? Okay, if you think of any, let me know. I am happy to answer your questions. In the meantime, I'm going to get another bacterium set up here. Okay, now back in chapter 5, it talked about horizontal gene transfer, okay? And I left that to your own study, but we are going to talk about one type of horizontal gene transfer because this is a good point to talk about it, okay? Sometimes in the process of assembly, if the DNA chromosome of the bacterium has been chopped up randomly into the same size pieces of nucleotide that we see for the phage, for the virus, okay? It will accidentally get packaged, okay? So here I've got 2 phages, okay, that have phage DNA in it. So when they come over here, and let's say this one lands here, okay? It will inject phage DNA into the cell, okay, and proceed to make more phages, okay? But with this capsid that does not have phage DNA in it, okay, it is no longer a virus, it is no longer a phage. But the capsid goes on automatic pilot, okay, and it will inject bacterial DNA into the new cell, okay? And so let's say this one, okay, just because it's a different cell, okay, it may say, hey, you know what? I've been needing that. I have a mutation in this gene. This is a good gene. So I'm going to replace my DNA with the DNA that was brought by this transducing particle. Okay, it's no longer a phage. It is a method of transduction. Okay. And that's how DNA from one cell can get moved to another cell. Okay. So this one would be a transducing particle. And this one would be a phage. Okay, any questions on that one? All right, there's one more thing I want to go over before I ask you questions. Okay, there are some types of phages that are what we call temperate or lysogenic. Okay. Let me get my E. coli going here again. Okay, so they have a very similar replication cycle as the lytic virus or phage that I just went over. Okay, so they attach, they inject their DNA, okay, into the cell. Okay, so they undergo penetration and uncoating. Okay, but instead of going straight into assembly, what they do is they will integrate themselves into the host chromosome. Okay. And when it does this, when it integrates itself into the chromosome, we now call this phage DNA that's a part of the bacterial chromosome a prophage. And it just hangs out. It doesn't start to make more virus parts. It pauses, okay, at this integration step, and it just hangs out. And so when my E. coli replicate, or excuse me, when they divide, okay, so this cell has undergone binary fission. It has divided. And in the process of replicating its DNA, okay, it has replicated the prophage. Okay, so let's get our chromosomes here. So this cell underwent binary fission. It had two daughter cells, okay, and they both have the prophage. Okay. And every time the cell divides, it takes the virus with it. And the whole goal of a virus is to make more of its genome. And so it's happy. It doesn't have to leave the cell. Okay. And that's why we call these temperate or iso. Getting the right spot. Genic. Okay, whereas the one that we talked about before is lytic because it lyses the cell. Okay. So some prophages just have phage DNA. Sometimes they have carried DNA from other host cells. Okay. So some of them carry toxins or antibiotic resistance. Okay, that may be of benefit to the bacterium. Okay. Now, what happens though is, let's say that this cell was exposed to a nasty chemical agent. Let's say that it was exposed to bleach. Okay, so we've got a lot of chlorine floating around here. Okay, and it damages the cell wall. Okay, starts putting holes in it. Okay, and starts to break up the bacterial chromosome. Okay, the phage says, whoa, time to get out. Okay, this ship is going down. Okay, so then it comes out of the chromosome, okay, and it goes into assembly, or excuse me, it goes into synthesis. So it makes more copies of the phage, okay, and this is where, when it comes out, sometimes it doesn't do a very good job, and that's where sometimes it will pick up some bacterial DNA, okay, and it will synthesize capsids, okay, and then it will go into assembly and it will cause the cell to lyse. So it undergoes release, okay, and it goes out looking for another host cell that it can turn into a provirus again. Okay. So it maintains the genes to make a capsid, okay, just in case, you know, the bacterial cell starts to die and it has to get out and go find a new host cell. Okay. Hey, and we're quite interested in these kinds of temperate viruses, okay, because like I said, they tend to carry these other bacterial genes with them from cell to cell to cell. Okay. All right, questions. Okay, I'm going to ask you a question. Okay, so I'm going to pull up a poll here. Okay, so a piece of bacterial chromosome has been mistakenly inserted into a phage capsid. Okay? The capsid is released into the environment and attaches to another bacterial cell. What is likely to happen next? Yeah, we got some good voting. Oh, we just broke 30%. And by the way, if you're not sure, remember this is anonymous. And also this is practice. This is how it works best for me to study, is to quiz myself. And if I get it wrong, I'm going to remember the right answer better next time. Okay, we're at 72%. Let's see. Oh, there we go, 77. Let's see if we can break 80. Oh, we're stalled at 77. Okay, tell you what, just for the sake of time. Oh, there we go. Now we got 83. Let's see if we can get 90. There we've got 88. We're almost to 90. Okay, say for the sake of time, we'll go ahead and pause. All right, the favorite answer was B. Bacterial DNA will be injected into the cell. Good job. So, tell me why that's the correct answer. No, we're pretty quiet today. Yeah, why? Why is B the right answer? The bacterial DNA will be injected. Yes, Amanda.
So I got it wrong. I picked C, but I thought that was because it's a, I don't know, maybe it's B because it's a bacterial phage, not just a phage.
Ah, okay. So it is easy to get those two backwards. Okay. And that's why I put them on there. I will do that on exams. Okay. And it is a phage capsid. Okay. But instead of having phage chromosome put into the capsid, we get bacterial DNA put in instead. So there's no phage DNA to get injected. Okay. Kind of see how that works? Yeah, because there's only bacterial DNA in that capsid or transducing particle, okay, it's no longer really a phage. Okay. It is a capsid, but it's carrying bacterial DNA instead of phage DNA. Yes, I've got 2 more hands raised. Michelle, yes.
How does that happen? How does the bacterial chromosome mistakenly get inserted?
It's because there are certain enzymes that just look for the right length of, actually they're not enzymes, they are proteins, so that look for the right length of DNA, and they don't check to see what DNA it is. So if it's just by randomness, that when the phage was chopping up the bacterial chromosome, they get a piece that's the right length, those proteins will grab it and stuff it into a capsid.
Does that have to do with them not having having a lot less proofreaders of whatever they're making, or is that different?
Oh yeah, so very good to bring that up. Okay, it is different. Okay, the proofreading is what happens with the polymerases, either RNA polymerase, which has less proofreading than DNA polymerase. Okay, and that's what during synthesis when more of the chromosomes are being made, more of the phage chromosomes.
Okay, so okay, thank you.
Yeah, so this is after the phage chromosome is made, okay? When the lytic phage, okay, injected its DNA into the bacterial cell, it went snip, 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 snip on the bacterial chromosome, basically killing the bacterium. It just doesn't know it yet.
Okay.
Yeah.
Okay, gotcha.
Kind of gross, huh?
Yeah.
Yeah, it's like those zombie viruses. You're dead, you just don't know yet.
Yikes.
Okay, does that make sense?
Yeah, that did for me.
Yeah. Okay, so if you pick C, don't worry, that was the next best right answer or the next best answer. Okay, it wasn't correct, but it showed that you were using some good reasoning. Okay. In this one, okay, that also was incorrect, but it was a good answer. Okay. Because it's showing that you're thinking. You're thinking, oh, there's no phage DNA inside, therefore it's not really a phage. Okay. So good reasoning. Okay. But the capsid does kind of act on autopilot. It just is set up to take the whatever DNA is inside, it's ready to take it to the next cell and put it in the next cell. Okay. And it doesn't matter whether it's phage DNA or whether it mistakenly got packaged with bacterial DNA, it just takes it on over. Yeah. Okay. All right, let us move on to animal virus replication, which is fairly close to bacterial replication, but we've got some important differences. Okay? Because animal viruses have a different structure. So animal viruses are huge. So I'm just going to draw a part of one, but we'll go ahead and get the nucleus over here. Because animal viruses are, or animal cells are eukaryotic. Okay. And we'll get some chromosomes going here. We'll just do 2 for the sake of time. If we were talking human animal cells, okay, because we are animals, okay, I would need to draw 46 of these puppies, which I'm not going to do. Okay. All right, so animal viruses have the same steps. So just for fun, what is the first step? What is the first thing that the virus has to do?
Attach.
Attach. It has to grab a hold, okay? So let's go ahead and do SARS-CoV-2. Just because that is what is on everybody's mind. Well, it's on my mind. I don't know about anybody else, but okay, so let's go get our crown of spikes here. Okay, it is an enveloped virus, which, so far as I know, no bacterial phages are enveloped. Okay. All right, so these spikes are embedded in the envelope, okay? And because it's an RNA virus, we're going to go ahead and put our RNA genome in there, okay? So the first thing that it has to do is grab a hold of the cell. Okay, so both phages and animal viruses will grab a hold of proteins on the outside of the cell that match its spike. Okay, so it's kind of like a docking station. Okay, so it attaches that way. And let's talk about specificity. Okay? Because most viruses are very, very specific into what kind of cells they will dock onto, that they will attach to. And it's because the shape of the host receptor has to match the spike. Okay, if I have another protein that looks more like this, okay, that's not going to grab a hold very well. Okay, so it is looking, so to speak, for these more round host cell receptors. Okay. All right, so they grab a hold, and what is the next thing that it has to do?
Penetration and
Penetration.
encoding.
Yep, penetration and encoding. Okay, now, because we do not have a cell wall, viruses can get in a number of ways. From what I was able to find out about coronaviruses, they can penetrate, okay, by having their lipid envelope fuse with the cell membrane. Okay. So that kind of looks like this. Okay, let's get rid of our cell membrane. I will put it back in. And I will leave a space down here for later stuff. Okay. And so we have spikes on the outside here. And then the capsid gets spat inside, okay, of the cell. Okay. Now the RNA, while it's in the capsid, cannot get, cannot make more of itself. Okay. It has to get rid of the capsid, okay, so that it can have its RNA out here, so that it can go to the next step, which is, what is our next step?
Synthesis. It breaks
Very
apart.
good. Yeah. It makes more parts. Okay. So this capsid has to break apart. Okay. So that's encoding. Now, COVID carries some viral specific enzymes. Okay. I'm going to draw them as kind of little pac-man guys. So one of them is an RNA polymerase that will grab a hold and will make more RNA. In fact, it makes mRNAs from the RNA genome. Okay, so this is the genome, and this is the mRNA. Okay, so the mRNAs get clamped onto by ribosomes, and the ribosomes proceed to make virus parts. Okay, so I make parts of the capsid, I make parts, I make some spikes. Okay, and these spikes get embedded in the cell membrane. Okay. Meanwhile, these parts here are going to start snapping together, okay, to make capsids. And then these mRNAs are going to be transcribed to make more genome. Okay. And the genomes get packaged. Okay. And my viral specific enzymes get packaged. Okay. And what happens is now that this capsid is packaged, all it needs are spikes in an envelope. Okay. So what it'll do is it will it'll convince the cell to move it to the cell membrane, okay, where its spikes are waiting. Okay, and we call this budding. Okay, so this snapping together, okay, is a part of what process? Assembly. Okay, and the budding, okay, is a part of the assembly and release process. Okay, so eventually this will pinch off, and I have a fully formed, ready to go SARS-CoV virus that is ready to infect the respiratory cell next door, or it can be coughed and sneezed out to infect another person. Okay. Okay, questions about how the virus does this. Okay, now in the textbook, it talks about how other types of animal viruses do these different stages of replication. Okay. And so it all depends upon what their genome is, okay, whether they're enveloped or not. Okay. They've got all sorts of ways of penetrating and uncoding, okay, and they have different ways of releasing. Okay, so I will leave you to take a look at that. Make sure you follow the study guide to see what I consider to be more important. Okay, and we have 2 minutes left, so we're going to go back to the PowerPoint. Okay. So I have some questions here that I didn't have time to get to, but I will make sure that they are in the practice quiz for chapter 6. Okay, so you can practice with these. Okay, so since we just have a couple of minutes left, let's go ahead and talk about what to do for next time. So I will have case study number 1 done by the end of the day on Sunday. Hopefully it'll be Saturday, but I make no guarantees. So as a reminder, what I want you to do is take a look at the answers that popped up in week 4. Okay. Take a look at that and compare it to what you did. Okay. And then when I get your assignment graded, you can click underneath the name and my grades. There'll be a little box that says rubric. And you can click on that and you can see which ones I clicked. Okay. And that'll show you, why you got the grade that you got. And if you want to go over it in more detail with me, then you can definitely come to my office hours and I'll go over your specific case study one with you. Okay. Also, now is a good time to complete the embedded tutor assignment number 2 if you haven't done so already. You can do that either by attending Rochelle's study sessions on Collaborate. Okay, the link is in faculty information. Okay, or you can go to the discussion board. Okay, and then exam 2 opens up this Friday. Okay, and it will be available until a week from Saturday, which is March 12th. Okay. All right, any questions? Yes, sure.
So on this case study 2, is it the same as case study 1 where we had to find resources and and answers through that direction like we did before?
Yep.
Okay,
Yeah, it's just this one fits in with with the genetics chapter and with this chapter because it talks about finding unique viruses by screening patient blood for unique genome sequences. Yeah.
so use of the textbook as well and so on. Okay, thank you.
Yep. Oh, quite welcome. Okay,
I didn't know how to get started on that one. So I don't, where do we begin? Okay,
Yeah,
thank you.
Yeah, with the case studies, it can feel like that. But it's the same process that we did for case study number 1. So hopefully because you've done one, it'll be less overwhelming. But, usually it takes 3 times before we start really feeling comfortable with a new assignment, with a new process. Okay, other questions? Yes, Amanda.
I don't know if you can probably answer this because I know you like to keep things a surprise, but I'm just wondering just exactly how much of chapter 5 will be on the exam, because that is a lot of like minute information to try to study. And it's just a lot. So I was just curious if you could
Yeah,
a little bit.
it is a lot. I agree. I've broken up the test into equal parts of each of the chapters. So a third of it will be chapter 4, a third of it will be chapter 5, and a third of it will be this chapter 6. Okay. What I would recommend is go through the practice tests, practice quizzes, okay? Because that will give you some practice in the harder application style questions. Okay. Sarago will help you learn the terminology that's important. And if you have the 2 ends, the understanding questions in the middle should come.
And also, so according to your schedule, it says the exam will be from chapters 1 through 6, but is it more like you're going to need to remember some parts from each of those chapters to answer the questions for 4, 5, and 6?
Yes, I'm not going to ask you any questions that directly relate to chapters 1 through 3, but you have to know the information in those chapters to do well in understanding chapters 4, 5, and 6. So, for example, today we talked about bacterial phages. You got to know the structure of bacteria, prokaryotes. Okay? We talked about animal viruses. You have to know the structure of eukaryotic cells. Does that help? Genetics, you needed to go back and review chapter 2.
Exactly. No, that helps tremendously. So, I was kind of freaking out that I had to keep remembering everything from chapter 1, 2, and 3, but knowing those little nuances will help me studying for this next exam.
Yeah, but it would be good to go back and just, kind of briefly skim those previous chapters. Hey, other questions?
That helps a lot. Thank you.
Oh, you're quite welcome. All right.